Hello, hello. Welcome, everyone. My name is Kevin Curran. I'm with Bridgepoint Bio, and I'll be recording these kind of short 3 to 5 minute videos for my newsletter subscribers where I explore a recent news story in advanced medicine. So we'll be focused on gene editing, cell therapies, gene therapies, and I'll be spending a little bit of time on the background science behind these medicines because I think that helps people appreciate the recent clinical commercial challenges that these drugs are experiencing as they move forward. So today we'll focus on gene editing, specifically base editing, a new tech platform of gene editing, and we'll be talking about it in light of a clinical hold that we just saw last week with Verve Therapeutics. So what's gene editing? Just to bring everyone in, this is a new drug category, a new biologic modality that relies on scientists using tools to make precise edits in DNA, in the human genome. And we've already seen the first gene editing drug approved earlier this year. That was Casgevy for sickle cell disease. So Vertex and Crispr brought that forward. Base editing, like I said, conceptually, it's a more precise way to go about making a DNA modification. Base editing changes one letter at a time. So a C to a T or an A to a G. And the difference here is that base editing, it still uses the guide RNA to find the area of DNA it's going to modify. But instead of a full Cas9 enzyme that creates a double-stranded break, base editing uses a modified or impaired Cas9 that only creates a single strand break. So just one break in the sugar phosphate backbone of a DNA helix. The idea here is that double-stranded breaks appear to be a little bit chaotic and could lead to unwanted effects. So a single strand break promises to be more precise. And the tech also works in non-dividing cells, which is a benefit as well for certain diseases. So a company named Beam Therapeutics has been moving a lot of this tech forward, and Verve Therapeutics in-licensed their base editing tech from them. Verve has been running a trial with Verve 101 for patients with high cholesterol, like a genetic form of high cholesterol, hypercholesteremia, and these are folks with much higher chances of cardiovascular disease. The approach here is to disrupt a specific gene in liver cells. So if you can make this single letter change in PCSK9, that should lead to a drop in LDL levels. That's the biomarker LDL, and that should drive forward the drop in cardiovascular. So that's the intention here. Verve 101, it's been in a phase 1 trial, so they're exploring different doses to see, early efficacy data and then see where any toxicities arise. You want to think about this medicine as two separate components. There's the delivery system and then the cargo, right? So in this case, the cargo is the gene editing tech, right? That's the guide RNA and the impaired Cas9 that's going to actually land on a chromosome and make a precise genetic change. And how is that all going to get in place, right? Because this drug is administered in vivo, so patients are getting an intravenous injection. You need to wrap up those tools with something to stabilize them and sort of help passage them into the right target cells. So the company Verve is using lipid nanoparticles, a specific proprietary LNP, similar to what was used to deliver the Moderna and Pfizer vaccines. So, data has come out on this Heart-1 trial. The good news, which is part of the message I want to deliver here today, is that the base editing technology appears to be working. especially patients in the higher dose arms, they saw significant drops in LDL levels, right? But the downside here, the problem is a safety issue arose. So there's 13 patients in this trial. There's 6 patients in the high dose trial, and one of those 6 saw a spike in liver enzymes. So specifically ALT enzyme and a drop in platelet counts. So that's a grade 3, serious adverse event. It was connected to the therapy. So that's a problem. And Verve has taken pause and decided to switch gears and switch to a different drug from Verve 101 to Verve 102. And I'm looking at quotes from the CEO. Verve is sounding very confident that the base editing technology is working, and that the safety issue was driven not by the cargo, but by the delivery system. And based on some animal work that they have with LNP with and without the base editing tools, they think it was the LNP carrier system that caused those safety issues. So we can zoom in and look at this lipid nanoparticle. It's a little ball of fat, right? These little lipids on the kind of the surrounding membrane of this particle. Verve is sort of tracking it back to this cationic lipid that was involved as a carrier for Verve 101. That's their opinion, right? And so what they've decided to do is switch to Verve 102, which is, as far as I know, it's a similar base editing system, but it's got a different LNP with a different cationic lipid. Okay, so again, this is sort of where the field is at. This is where the field is at with gene editing. The tools appear to be working. We're still kind of moving in on higher levels of precision, accuracy, minimizing on-target, off-target, unwanted effects in terms of editing. But this delivery piece is a challenge because liver cells are one of the cells that we're most proficient at in terms of hitting in an in vivo administration. So when we inject LNPs into the bloodstream, liver tissue is one of the easier cells to hit. So I think this trial just highlights that we're not out of the woods yet. We still need to, iterate and optimize these delivery systems if we want to be able to deploy these gene editing tools to all these different cells in the body. Okay, folks, that's a wrap. That was plenty of information. And feel free to comment and let me know any other topics you'd like me to touch on in the future.